The overall goal of this procedure is to build a regression model for the prediction of malaria parasitemia using ATR-FTIR spectroscopy with multivariate data analysis.
This method can help expedite point-of-care diagnostics, which can help improve patient outcomes in the future. The main advantage of this technique is that while the model may take some time to build, it is highly portable, user-friendly, highly sensitive, and low cost.
Though this method enables the quantification of malaria parasites, it can also be used to observe phenotypic response to environmental changes, such as changes in parasite composition due to antimicrobial agents.
The idea developed from work we did at the Australian synchrotron on malaria-infected cells. This was at the single-cell level. From there, we decided to use ATR-FTIR spectroscopy to actually develop the diagnostic test.
Prior to starting the synchronization procedure, culture 30 milliliters of 3D7 strain Plasmodium falciparum parasites as described in the text protocol. Using an automated pipette, resuspend the culture and transfer it to a 50 milliliter conical tube. Centrifuge the culture at 300 to 400 times G under standard laboratory conditions for 5 minutes. Remove the supernatant by drawing it up with an automated pipette without disturbing the pellet. Discard the waste media into a 10% bleach solution. Slowly add 12 to 15 milliliters of a 4% sorbitol solution to the pellet. And mix the culture by